Hey friends, welcome to this channel. If you're new here, I am Indrani from Bong Biology, and in today's video we'll be discussing about endoplasmic reticulum and how glycoprotein is folded within the endoplasmic reticulum. This is, I think, the fifth part of this endoplasmic reticulum series, and hopefully this is the last part also. So we'll just summarize what we learned in the previous video about the folding of the glycoprotein. We learned that first the N-linked glycosylation will happen co-translationally, and after that it will enter into the endoplasmic reticulum lumen, and glucose molecules and all these oligosaccharides will be linked on the asparagine residue. Okay, so this is a receptor asparagine residue, and then it will enter into the calnexin where the protein folding will occur. If the protein is folded correctly, it will exit the transitional endoplasmic reticulum. However, if the protein is not folded correctly, it will again enter into the calnexin where glucose will be added, and in this second cycle, if the protein is folded correctly, it will exit the transitional endoplasmic reticulum. But if it is still not folded correctly or unfolded, then all these mannose residues will be removed, and then it will be translocated back to the cytosol where there will be polyubiquitination of this protein, and so it will enter into the 26X proteasome and will be degraded. Since this ubiquitination and polyubiquitination is ATP dependent, so one ATP will be converted into ADP. But what happens in case of unfolded protein? Okay, now normally what happens is the concentration of BIP in the endoplasmic reticulum lumen remains so high that it can bind with the unfolded protein. Also, it can bind with several other signaling molecules. But what happens if the cell is under stress? Under stress means there are lots and lots of unfolded or misfolded protein is present within the endoplasmic reticulum. Then in that case, under cell stress, what happens is all this BIP protein that were initially or normally associated with the signaling molecules will be removed from the signaling molecule, and they will bind with the unfolded proteins. As a result, this signaling molecule will give some sort of signal, and that is called unfolded protein response. And as a result, what happened is more and more BIP will be produced, more and more proteasome will be produced, more ubiquitination of the protein will be done. So ultimately all this misfolded or unfolded protein will get degraded. Okay, so this happen in case of cell stress. This is in case of normal protein folding where BIP is associated with the signaling molecule. BIPs are also associated with some of the unfolded protein, and most of the proteins are folded correctly. But under cell stress, all the BIPs are concerned with the unfolded protein, so there is unfolded protein response, and as a result there will be production of the BIP concentration will be more, and then the proteasome production will be more, ubiquitination or what we called is polyubiquitination, this will be more, and as a result finally there will be more protein degradation. All the unfolded or misfolded proteins will get degraded. Okay, now next we'll discuss about the smooth endoplasmic reticulum and lipid synthesis. So all of this thing we discussed was about rough endoplasmic reticulum, and now we'll discuss about the phospholipid synthesis. Phospholipid synthesis in smooth endoplasmic reticulum. Now hope you can see the figure. Glycerol phospholipids are synthesized in the endoplasmic reticulum membrane from cytosolic precursors. Two fatty acid chains are linked with the coenzyme A. These two, they carries and first join to the glycerol 3-phosphate. So both of them, they are first joined with the glycerol 3-phosphate. As a result, the 2-CoA residue will be removed and it will produce phosphatidic acid. This phosphatidic acid can be converted into diacylglycerol, and through CDP choline it can be converted into phosphatidylcholine. This phosphatidic acid can again be converted into phosphatidyl inositol, and from this diacylglycerol phosphatidyl ethanolamine and phosphatidyl serine can be produced. So the thing is, we have two fatty acyl CoA and we have one glycerol 3-phosphate. Two CoA group will be removed and two fatty acid group will be added to the glycerol 3-phosphate. So that's why this is called glycerol phospholipid. Phospholipid means there will be fatty acid and phosphate, but this is glycerol phospholipid because this is the precursor is glycerol 3-phosphate. Okay, hope that makes sense. We have glycerol 3-phosphate, we have two fatty acyl CoA, and these fatty acid residues are being associated with the glycerol 3-phosphate, so that's why we call it phosphatidic acid or glycerol 3-phosphate. But if you can see over here, every time the glycerol part or the fatty acid part is joining, it is joining on the outer phase, that means at the cytosolic phase of the endoplasmic reticulum membrane. This shouldn't be done because if this continues, then there will be no membrane bilayer equilibrium will be destroyed. So we have another protein which we call is flippase. What flippase does is, we have the phospholipid bilayer, and what flippase will do is it will flip this phospholipid bilayer to the opposite side. So let's say this is the phospholipid glycerol phospholipid bilayer, and it will flip this to opposite side so that there is an equilibrium of glycerol phospholipids on both sides of the plasma membrane, or of the endoplasmic reticulum membrane basically. And finally how these proteins which are being processed and sorted within the endoplasmic reticulum, how they come to the plasma membrane or their final destination is by vesicle budding. This vesicle budding, that means formation of vesicle, always happens from the transitional endoplasmic reticulum, and then they are joined with the ERGIC, that stands for endoplasmic reticulum Golgi intermediate complex. So endoplasmic reticulum Golgi intermediate complex, they are present in between Golgi apparatus and endoplasmic reticulum. So they first enter into the ERGIC complex, and from this ERGIC all these proteins are carried to the Golgi. And luminal endoplasmic reticulum proteins are taken up by the vesicles and released into the lumen of the Golgi, and membrane proteins maintains the same orientation in the Golgi as in the endoplasmic reticulum. So membrane protein will be transported to the membrane of the Golgi, and cytosolic protein or luminal protein from the endoplasmic reticulum will be transported into the lumen of the Golgi. So this is how the whole protein sorting things happen. I hope you can understand, and if you like it, please make sure to like, share, comment and subscribe to my channel. And you can also follow me on www.bongbiology.co.in, this website, and this website also contain all my contact details. So yeah, that's it. Hope you enjoy, and good night.